Alright, Assalamu alaikum everyone. My name is Abdulrahman Alabyad, and inshallah today I'm going to be explaining to you the MOL lecture of cellular protein sorting and targeting. So first of all, I just want to wish you guys the best of luck on your CVP grades. And so yeah, I don't want to take much of your time. I know you guys are tired, so let's finish up this quickly. Anyways, here's my contact number. If you guys want to text me later on whenever you study the lecture. If you have any questions, feel free to text me whenever you want for whatever you want. Okay, so to begin, what is the main focus of this lecture? Basically, protein sorting and targeting. All right, so, okay, so main thing we talk about in this lecture is that what are proteins and where exactly we need these proteins. So in order to make it easy, let's go back towards the basics, all right? So what are proteins? Why exactly we need proteins? And if we understand why we need them, then we'll know where exactly we're gonna need them, right? So right here, I brought for you guys a bunch of examples for different functions of proteins. So on the top left-hand side, we have one of the functions, which is transport. And an example of these proteins is hemoglobin, as you can see here. Now, can you guys tell me what exactly is the function of hemoglobin, or where exactly is it transporting? Anyone in the chat, or you can unmute your mic. Exactly, so as simple as that, hemoglobin is just transporting oxygen. And because hemoglobin is transporting oxygen, where is it going to be? Where do we suspect to find it? We're going to find hemoglobin in the blood. So one of the functions is transport, and one of the destinations we might need it is the blood. Another thing is you got this receptor right here. See, this is a normal membrane of the cell, and then you got this large thing that we call a receptor. So this is another protein. And the proteins shown in this picture is the sodium-potassium ATPase, one of the most important, if not the most important and most abundant receptor on our membranes. Okay, so 3 sodium out, 2 potassium in. Again, this is just an example of another function. So far we got 2 destinations. We got the blood and we got the cell receptors or the cell membrane. Another protein we got is hormones. So right here in this picture, we got this protein known as insulin. And what is the function of insulin? The function of insulin is basically whenever you eat a meal where you have a lot of sugar in your body, this insulin is going to be produced by the pancreas to lower the insulin level. But this is not part of the lecture. What we need to understand from here is that hormones is another protein function, and we need them for the blood because hormones circulate throughout the blood. And lastly, we need them for enzymes. And this is probably the most important thing, enzymes, because enzymes are basically helping you carry out everything in your body. So for example, right here is a picture of a protein. See this orange thing? It's a protein. And it's an enzyme that is going to be used in this process of transcription, I think. So basically, if you don't have a protein or protein enzymes, you can't make other proteins and you can't keep the cell alive. So this is why enzymes are considered one of the main and most important functions of proteins. With that being said, where do you expect to find enzymes most commonly? Enzymes are going to be found inside the cell. And to sum it all up, we ended up with 3 locations for proteins. We need proteins in the blood or around the body. We need proteins inside the cell membrane, and we need proteins inside the cell, right? So if this is understood, the next part or the entire lecture today is going to talk about how exactly does the body know whether to send these proteins outside the cell to the body, to the blood, or to put them into the membrane, or to keep them inside the cell, right? So as you can see, we need them inside the cell, outside the cell, and on the cell membrane. How will the body know exactly where is it going to be sent out? All right. Before we go into that, there's just a small basic concept I need you guys to understand, okay? All proteins, where are they going to be synthesized? In the ribosomes, okay? So you got the process, the central dogma, and the entire MOL, first part of MOL, it says that you got DNA, which gives you RNA, and so once you form the DNA to RNA, RNA gets sent out to the cytoplasm, and from RNA you get your proteins, okay? And so anyways, proteins. This process of RNA to protein is done by ribosomes, okay? So if you guys get that clear in your head, ribosomes are responsible for formation of protein, which I think you guys also took in MOL 1. Anyways, just keep this in mind whenever we're going into the process of cellular targeting, all right? Now, ribosomes, we have two different types of ribosomes inside the cell. The first one are the free ribosomes, which are found inside the cytoplasm. All right, so these ribosomes are synthesizing proteins and they are found inside the cytoplasm. So this is one thing. And another place where you can find these ribosomes are on the endoplasmic reticulum. So we got two, we got the smooth and we got the rough. The rough is because it has ribosomes. And so with that being understood, we can like suggest that the endoplasmic reticulum or the rough endoplasmic reticulum is helping in synthesizing some proteins, while the free ribosomes are helping in synthesizing other proteins, okay? Now, one last thing to know is that basically, what is the difference between the free ribosomes and the ones found in the endoplasmic reticulum? The difference is that this endoplasmic reticulum is going to synthesize the proteins that are going to be sent outside the cell, right? Is this clear? Just remember this. The endoplasmic reticulum synthesizes proteins to be sent outside the cell. So this is why it needs its own ribosomes. And the free ribosomes synthesize proteins inside the cell. And this is why they're going to be found in the cytoplasm. All right? Now, with that being said, as you can see here, the proteins synthesized inside the cell, they're going to be made inside the cytoplasm by the free ribosomes, and then you have them, you got your protein, whatever protein you made, it's inside the cell. You don't need to do anything else to it because you just made it and it's there. And which ribosome did you say do that? The free ribosomes. On the other hand, we got the proteins which are to be made for the membrane and to be sent outside the cell. We said these are going to be done by the rough endoplasmic reticulum wall, or the ribosomes on the rough endoplasmic reticulum. And are you done? If you have them inside the endoplasmic reticulum, are they outside the cell now? No, there is still a few sequence of steps to send them outside the cell. And this sequence of steps all takes place in something we refer to as the endomembrane system. So, to summarize, what is the endomembrane system? The endomembrane system is a system that starts from the endoplasmic reticulum and ends at the cell membrane, and it is responsible for everything that goes into the cell or outside the cell, okay? Keep this in mind. The endomembrane system is responsible for everything that goes into the cell or outside the cell. And one last final point is that, so you see how we have free ribosomes and the ones on the endoplasmic reticulum wall? Whenever you have any proteins to be made, it always starts on the free ribosomes. Okay? Any type of protein transcription starts on the free ribosomes. And then you so you might get a question, like, or not that an exam question, but I mean, you might ask yourself, if it starts on the free ribosomes, then like, why is the rough endoplasmic reticulum there? Why are the ribosomes there? And like, how exactly does it get there? Basically, what happens, and we'll go into the details later, but what happens is that it starts on the free ribosomes and then it stops. It's not completed yet. Midway, it stops. It goes to the endoplasmic reticulum and then it hands it over to the rough or to the ribosomes inside the endoplasmic reticulum. And then the endoplasmic reticulum ribosomes, they're the ones that continue the process. Is this clear? This is all just like the introduction. I know I might have said a lot. However, like if you look back into it, it's just a few points. But are they clear to you guys? Do you have any questions before we keep going? All good? All right. So now, with that being said, we can go. All right. So here's another summary of what I said. So the ribosomes are making the proteins, and then the membrane system is basically going to be made of stuff that take endosomes, which basically take stuff outside the cell or into the cell. And what are the components of the endomembrane system? You got the rough endoplasmic reticulum, which sends them to the Golgi. Now we're talking about the proteins being sent outside the cell. So we got the rough ER, sends it to the Golgi. And then from the Golgi, you got this vesicle, which we refer to as the endosome. And then from the endosome, it's going to be sent outside the cell. And if there is a vesicle fusing with the membrane, so basically like a vesicle that fuses with the membrane, we call this process exocytosis. Cytosis because it's kind of part of the cytoplasm, cytomembrane, and exo is because it's going out. And with that being said, what would you expect to call it if it's fusing with the membrane to go into the cell? What do you expect to call it? Someone in the chat or someone on mute. If this is exocytosis outside the cell, then the one into the cell is called endocytosis. Okay, so it fuses and goes into the cell. All right, so with that being said, and once this is clear to you and the basic idea is there, now let's dive into the lecture. How exactly, so this basically summarizes the entire title of the lecture. How exactly will the body know if this protein is going to be made to stay inside the cell, or it's going to be made so that it goes to the endoplasmic reticulum ribosomes and to be sent outside the cell? So would any of you would like to take a guess what exactly would tell the body? All right, so it's the DNA. The DNA does help by synthesizing the sequence that we'll need, but I just wanted a simple word. It's a signal. A signal tells the body that it goes here or there. All right, so it's a signal. This is basically what we'll be discussing. We're talking about signals. Now, this is from your slide. It's basically the codes, right, or the signals that we have inside the cell. As you can see, a DNA gives you the RNA, and this RNA is used to get you your amino acids, right? And amino acids will make proteins. Now, a sequence of amino acids will create a signal. And so, just because you said DNA, I'm just going to say this. So, amino acids, they go back to RNA, which goes back to DNA. So technically, DNA is right because DNA sends out these amino acid signals to direct them. Now, there's a different signal for every location you might find proteins. So for example, if you want to send it to a nucleus, you have this signal. If you want to take it outside the nucleus, you'll have this signal. If you want to take it to the mitochondria, you have this signal, and so on. Basically, this is a list of the different signals. And I'm pretty sure, I don't think you guys need to know the signals. What exactly are the signals? However, do please make sure with your doctor and your 15th batch students. So just to be sure and to be on the safe side. However, there are two important signals that I will mention for you guys to remember. And right here, you got the breakdown. So basically, any green is negatively charged amino acids, and any positively charged are pink. This is basically your breakdown. Now, what are the two important signals that I need you guys to remember? The two signals are the one that sends it to the peroxisomes and the ones that send it to the endoplasmic reticulum. Now, why exactly I want you to remember the one that sends it to the peroxisomes? Honestly, it's just because the peroxisomes are mentioned in your slides as a later part that we'll discuss. So I guess you have to remember it. Okay. Now, however, now I want you guys to answer. Why do we need to know the signal for sending into the endoplasmic reticulum? Any guesses?
So that when the mRNA gets attached to when the free ribosome attaches to mRNA, it sees this code to know that it should be sent to the ER and not made in the cytoplasm.
Exactly. So that the endoplasmic reticulum is going to be, or the free ribosome sees the signal and knows that this protein is going to go to the ER. And as we said earlier, anything that goes to the ER is to be directed outside the cell. So in other words, all these signals that, so all the signals you see here are going to be for taking stuff or for making proteins inside the cell. And this is the only one that is responsible for proteins going outside the cell, right? And this is why it's important. Now, what exactly is it? It's basically a series of hydrophobic signals or a series of hydrophobic amino acids. So I made a mistake here. I should switch the colors. White is actually hydrophobic and blue is hydrophilic. So basically, look at the text and ignore the colors because I made a mistake here. Okay, so yeah, it's basically a series of hydrophobic amino acids, right? What is it again? It's a series of hydrophobic amino acids. And it is also another thing about it is that it's found on the N terminal. So this is an N terminal. I'm gonna go to this slide for a second. Now you got two terminals of a protein sequence. You got the N terminal, which is the start, and then towards the end you have the C terminal. So N terminal is the start, C terminal is the end, and in between you have your entire protein sequence. All right. So this is not important for now. All I need you to know is that it's found on the N N-terminal, and it's a hydrophobic sequence. Now, what is a way to remember that it's found on the N-terminal and that this is hydrophobic sequence? Basically, like to me, the way I remember it is that, it's a way our body is nice to itself. If you want something to be sent to the ribosomes that are found on the endoplasmic reticulum wall, why should your free ribosomes go through the mess of preparing them and making the entire thing just to find out later on that it needs to send it to the endoplasmic reticulum? Instead, as soon as it starts, it tells it, please, I don't want you, I want to go to the endoplasmic reticulum. So this just makes it easier. So from the start, instead of going through the entire mess and the work, from the start, you just know to send it to the endoplasmic reticulum. All right, so this is basically a summary of everything we said. As you can see, it's just a few points, nothing important probably. And now we're going into the big stuff. All right, are you guys ready? Get your water, drink a cup of water if you want. And now we're getting into the real stuff. What happens when we get the signal and how will the body respond? Now, in all the different organelles that we'll discuss in this lecture, there is a common theme, all right? So you got this protein. As you're making the protein, or once you're done with the protein, this sequence, it has a signal, all right? Every protein sequence has a signal. And now this signal, the only way to recognize it is by having a receptor. So a signal binds to its receptor. The receptor takes this protein from the free ribosomes or from the cytoplasm, takes it to the destination, all right? So one thing to note that if it wasn't completed, so for example, if we're talking about a protein to be synthesized in the plasmic reticulum, once the receptor binds, it pauses the transcription. So you can think of it as like some type of officer or like, some type of officer that's looking over this process. And, when someone is looking at you, you're just being shy, you don't want to do anything, you don't want to move. So this is basically what happens. So receptor binds, it pauses the process. The receptor takes it to the destination. And now, in order to send it outside the destination to send it inside into the destination, you'll need energy because you're pushing it. So pushing needs energy. And this energy is going to be gained by GTP or ATP breakdown or hydrolysis, right? So once you break down ATP, this does two things. It pushes the protein inside the cell and it gets rid of the receptor so that the receptor can go back into the blood or to the cytoplasm, right? And since you remove the receptor, if it was then the plasmic reticulum, you can continue the transcription. And so the protein enters into the cell by the push of energy through a channel. And then once it's inside the channel, you don't need the signal anymore. So you cut the signal off and you got your protein in its destination. I hope this is clear. Basically, I'm gonna leave it on this slide one more time so that you guys can remember it. And if you guys remember it right now, we're gonna use it throughout the entire lecture, and the lecture will be the easiest lecture for you. All right, so we got a signal that binds on its receptor. Receptor takes it to the destination. And then once there, it uses energy to push it into the cell through a channel, and then we cut off the signal. And the receptor, on the other hand, it goes freely. Right? Is this clear? Is this good to go? All right, one of you asked me to repeat again, so I'll do that in a second. Basically, we're going to repeat this now 5 different times for every different organelle, all right? So if you don't understand it for now, however, I do want you to understand this because it will help you a lot. I'm going to go over it again. So you have a protein which has a signal. This signal is responsible for directing us. And how do we find the signal? Through a receptor. So the receptor for the signal binds on the signal. And once it binds, everything pauses. The receptor takes it over to the area, whatever, let's say it's the nucleus. It takes it to the nucleus. Once it's inside the nucleus, the receptor uses energy. Why do we use, or the nucleus, actually, there is another receptor on the nucleus. So this receptor on the nucleus will recognize this compound, and so it binds on that nucleus receptor. There, the nucleus is like, why are you touching it so tight? It uses energy to separate them. And if they're separated, the nucleus takes the one that's getting bullied, and the bully gets to go back to the cell. And so the bully, or the one bullied, which is the protein, the nucleus takes it in, pushes it inside of it, and that's it. You got your protein inside the cell. All right? Is this clear? So you can think of it like a bully, which is a receptor, and bullying the signal, and then whatever organelle we're going to is basically like the teacher separating the two bullies. If you understand it that way, It makes it very easy for you. All right. Now let's go to the first thing, which is proteins to be made outside the cell. And if you remember from the first slide, where did we say we need them? We said we need them on the cell membrane, we need them inside the blood to be secreted, and this thing, we didn't mention it, but we also need them in the lysosomes, all right? We need them in the lysosomes as well. Now, please, guys, if I go a little too fast, please feel free to let me know in the chat, and I'll try to slow down if my pace is a little too fast. All right, let's begin. First thing we have is the rough endoplasmic reticulum. Oh my bad. Okay, so I should have asked you this. Proteins to be sent outside the cell, where will they be made? In the cytoplasm or in the endoplasmic reticulum? Where are we going to have the proteins to be synthesized outside the cell? Is it the rough ER or is it for the cytoplasm? Anyone? Okay, so it's the rough ER. Thank you. All right, so now we said, you guys ready? Let's go to the endoplasmic reticulum. We said every process starts in the free ribosomes. So we got our free ribosomes. They start the translation. Then we got our signal, right? Signal, first thing that happens is the signal. I'm going to use the theme that we had in the previous slide for everything, all right? So first thing that happens is that the signal appears, or the person that's about to get bullied. Now, what did we say was the signal for anything sent outside the endoplasmic reticulum? Say it with me. It's the N-terminal hydrophobic signal sequence, all right? It's the N-terminal hydrophobic signal sequence. Now, once you have the signal, you got the receptor of the signal, and we refer to it as signal recognition particle. It binds on this signal. So now it's bullying the signal. So the translation is paused. Okay? So once it's binding on it, it takes it with it, it's going around, it takes it to the endoplasmic reticulum. Now, the endoplasmic reticulum is our teacher. It has a receptor for the signal. So there's a signal recognition. So this receptor part, there is a receptor for the signal recognition particle, or for this complex. So this receptor on the endoplasmic reticulum, I put alpha chain because basically it's two subunits, alpha and the beta subunit. The alpha subunit is the one that recognizes it, okay? So the endoplasmic reticulum recognizes this compound. And what does it do? It uses energy to separate it. So we hydrolyze GTP. Once they bind, both of them will hydrolyze GTP to GDP. And when you use this energy, you get rid of this signal and you put the protein inside the cell. Now, what is the channel that pushes the protein inside the cell? It's called translocon, which you can see it right here. It basically opens to pushes the protein inside the cell. And then since we now removed the signal recognition particle, inside the cell, we can undergo transcription, or we can continue a process of translation in the rough endoplasmic reticulum ribosomes. Lastly, once it's inside the cell, we have a signal peptidase to remove the signal because we no longer need the signal. And let's assume, to go back with our analogy of the bullying, once you remove the signal, it's no longer young, it's no longer weak, it's now strong and mature, and basically it doesn't need anything else to support it, right? So now it's a big protein, right? So now you get it. Hold up, let's give me my mouse. So you remove the signal and you can complete your process of translation. Is this clear to you guys? Is it easy? All good? Any questions? All right, inshallah, it's all good for you. So now the next thing that we need to is basically, remember how we said that once it's inside the endoplasmic reticulum, we have this entire sequence or like this entire system that is responsible for helping us release these structures. And we said it's called the endoplasmic membrane system. Okay. Now, what exactly does the endoplasmic membrane system do? Please ignore the text. I should have hidden it so that I explain a bit. So please ignore the text and just focus on what I'm saying now. So this system, what it does is that once it takes the protein inside of it, it will modify it, it will fix it a little bit. And once it's fixed, it will fold it, package it, and send it out to the Golgi apparatus, or send it out to the blood, or to the membrane, or to the lysosomes, okay? So what does it do? This system modifies, packages, and sends it out, okay? So one of the important modifications that the protein undergo inside this endoplasmic membrane, and like it was mentioned in 2 slides for you guys. So one of the most important ones is basically addition of a sugar, right? And a protein with a sugar is referred to as a glycoprotein, or you have another type, the proteoglycans. Whether, like, if it has more sugar, then it's a glycoprotein. If it has more proteins, it's a proteoglycan. Okay, so glycoproteins is one of the most important modifications. Is that clear? All right. Now what happens is that it is going to be modified, folded, and sent outside the cell. Okay, and you can have other modifications inside the Golgi. The folding process is done by chaperones, and once you send it outside the Golgi, you go with endosomes. Okay. Now, one last point is that what happens if you have failure of folding by the chaperones? Folding of the protein inside the endoplasmic reticulum is our important signal for telling the cell or telling the endoplasmic reticulum that once you fold it, you can send it to the Golgi. If you can't fold it, you can't send it to the Golgi. All right? Keep this in mind. Folding is the signal to send to the Golgi and outside the cell. Okay? So if you fail to fold it by the chaperones, then you have a defect in this protein, which is basically going to be something we'll discuss later on. But just for now, no. If you have failure of folding, it will cause a problem in the proteins, and so you break down the protein, right? So we said the modification, one of them was glycosylation. Now there's 2 types of glycosylations that you guys really need to know and differentiate. The first one is N-linked glycosylation. Now what happens in the N-linked, basically it is forming or taking place in the rough endoplasmic reticulum. It's basically adding an N-acetylglucosamine sugar on asparagine residues, or basically see as the asparagine right here. We add, oh my god. Okay, so yeah, so on asparagine, you add this sugar and acetylglucosamine, and it happens before folding. Why is it happening before folding? How to remember that? Because it's in the endoplasmic reticulum. And we said the endoplasmic reticulum is responsible for folding. So if it wanted to modify it, it can modify it before folding it. Okay? So just remember these. And one way to remember that N-linked is associated with asparagine is using this N that is found in asparagine. Okay? All right. So this is one thing. Other type is O-linked glycosylation. Now, O-linked takes place in the Golgi, and I'm going to underline the O because it helps you remember. O is for Golgi apparatus. It occurs on either serine or threonine, and no longer asparagine. Okay, it's serine and threonine. And the sugar added is N-acetylgalactosamine. So in N-linked, we had glucosamine. Here we have galactosamine. Okay. And lastly, it occurs after the folding of protein, because remember, what did we say was the signal of sending a protein to the Golgi? It's the folding. So once it's folded, it's in the Golgi, and so it's the Golgi receives it folded, it will add the sugar onto a folded protein. Okay, so please remember these 2 different types and know how to differentiate. Now, one last special type of protein that has different modifications, and not just glycosylation, it is basically collagen. Okay, so now, I need you guys to remember the sequence first of all, and then understand the process of that, or basically the modification that's happening. Now, the sequence that happens is that once you have basically the DNA to RNA, you go, since it's going to be secreted outside the cell, where do we take it? We take it to the rough endoplasmic reticulum. Now, the sequence is a pre-pro collagen, or basically any proteins that have multiple sequences, we refer to them as pre-pro protein or pre-pro collagen. Then we remove this pre. So what do we have now? Pro collagen. And then we remove this pro. Or before we remove it, basically, we're going to add a tro. We add a tro to it. So now it's a tropo collagen. And then once it's done, we remove this tropo and we got our collagen fibers. Okay. So it's a pre-pro collagen, pro collagen, and usually it should be collagen right after that. However, there is this small step of something we refer to as a tropo collagen. Okay. Pre-pro collagen, pro collagen, tropo collagen, and collagen. Okay, so first thing we said is that we're going to take it to the rough endoplasmic reticulum, and there's going to be modifications. So we said the main modification that happens is glycosylation. However, before the glycosylation, there is this small modification known as hydroxylation. Okay, so what is hydroxylation? One second. Okay, apologies about that. So what is hydroxylation? Hydroxylation is basically the addition of a hydroxyl molecule. All right. So very, very high yield to remember. Very high yield. I highlighted it even in red. Hydroxylation requires vitamin C. Okay. So you might get a question. Which of the a person that has vitamin C deficiency, which of the following steps is disrupted? It's always hydroxylation. All right. And okay, so yeah, basically hydroxylation. Now, the next thing that we got is glycosylation. And once you have the glycosylation, you now got your pro-collagen. The pro-collagen here this time is going to be a triple helix. Why is it triple helix? Because we got 3 different chains that we're going to rotate them around each other, so they're going to form a triple helix. All right? Now, so a pre-pro-collagen undergoes hydroxylation and glycosylation. Okay, and there is a mnemonic to remember, like hydroxylation and glycosylation, like which comes first. It's basically like S, H, G. That's how we used to remember. I used SHG. Synthesis, hydroxylation, glycosylation. Okay, so S stands for synthesis, hydroxylation, glycosylation, SHG. Okay, so pro-collagen is going to be sent into the Golgi apparatus, and from the Golgi, it's going to get further modified, and then it's going to get sent outside the cell. So once it's outside the cell, what happens with the collagen? We remove the ends, we cut off the terminal ends. It has its own haircut. Once it's old and mature, we send it out, we give it a haircut. We cut off the C end and the end terminal. And now this is going to be referred to as a tropocollagen. And this is also another very high yield thing. Why exactly do we need to cut off the terminals or the ends? Because we want it to become insoluble. Where do we have the collagen? We have collagen in the cell membrane. And in order to make it insoluble, so that, or not in the cell membrane, okay, my bad. We have the collagen in the cellular matrix, which is between the cells, outside the cells. In order to avoid having it get dissolved in the fluids there, we need to make it insoluble. And this is done by the cleavage that happens at the terminals, okay? And lastly, you need to make it stable and strong. And this is done through the process of cross-linking. And another time, very high yield, that is found in red. The enzyme doing the cross-links is lysyl oxidase, and it requires copper. Okay, so it's the lysyl oxidase enzyme that requires copper. So again, just like the vitamin C deficiency, you get another patient. This patient has copper deficiency. If a patient has copper deficiency, then it's a problem with the cross-linking. They're not able to form a stable, strong collagen particle. The collagen itself is fine. However, it's a weak collagen because you're not able to have a strong one by the lysyl oxidase. Okay, so like you might get this hint or this in the question, is the problem. So try to understand if the problem is in the collagen structure itself or in the strength of the collagen wall. So yeah, if it's a problem in the cross-linking, it's a problem in the collagen wall. I'm gonna pause this 1 second. I'm gonna due to my background. Sorry about that. Thinking in the sound. Okay. Yes. Sorry about that. Okay. So we were right here. Okay. So is this clear? You guys understand the collagen part? All good with collagen? Do you guys make sure to remember the high yield stuff? Any questions? Please let me know. All right. Okay. As we said, modification is done, and we sent. Okay, so yeah. Now modification is done. We sent the proteins outside the cell. So now, what did we cover? We covered 2 things. We covered the proteins to be sent outside the cell, and the proteins to be placed inside the cellular membrane, because we sent them out of the protein system, and they can, just like collagen goes outside, the protein can attach on the cell membrane. The only one left is the ones that are going to the lysosomes, right? So this is a new one. We didn't discuss the lysosomes. So what exactly happens in the process that goes to the lysosomes? Okay. Now, what exactly happens? So you got your protein that we put in the endoplasmic reticulum. So we said in the endoplasmic reticulum, you're going to have modification. And this modification was glycosylation, and it was end-linked. The O-link occurs in the Golgi, so the end-link. Now, the key point here is that the protein or the glycosylation that occurs in the lysosomes, it has a sugar known as mannose, okay? Please remember this. Mannose is the sugar that is going to be added. Then mannose is the sugar that's going to be responsible in the end-link to glycosylation. I know that we said it's an acetylglucosamine, but what I mean exactly is that this N-acetylglucosamine, the one part that is going to be added in for this case is going to have a lot of mannose, okay? So you have the N-acetylglucosamine and a lot of mannose that's going to be present on it, okay? So both of them. Now you got a lot of mannose. Now, so we modify it, we fold it, we send it out to the Golgi. Now this time it's a little different for the Golgi because it sees mannose, all right? So this mannose is a new thing. What does the Golgi do? It basically tries to poke it because it's a new thing. You know, whenever you see a new thing like a child, try to poke it. So this poking is basically the Golgi apparatus adding phosphate. So it's basically phosphorylating this mannose. And what is the enzyme responsible for this process? It's N-acetylglucosamine 1-phosphotransferase. Why N-acetylglucosamine? Because we said it's an N-linked glycosylation. And what is phosphotransferase? It's the transfer of a phosphate particle. So this is basically the modification that happens in the Golgi. Now that you got the mannose mannose, which is phosphorylated, this mannose phosphorylated is now going to act as a new signal, which has its receptor. So the receptor takes this mannose 6-phosphate, and this time, since it's in the Golgi, it takes off the Golgi as an endosome. We said always after the Golgi, you got the endosome, right? So it takes off with this mannose 6-phosphate on this receptor inside an endosome, okay? And what happens now is that we need to get rid of the stuff that we added to this sugar. Everything the Golgi added, we need to remove. So what did the Golgi add? The Golgi added the phosphate, the Golgi added the receptor. How do we remove the phosphate? To remove the phosphate, you bring an enzyme that cuts off the phosphate, which is a phosphatase. You see the tase? Tase basically is removing, or anything associated with removing has this ase. So phosphatase removes the phosphate. And how do we remove the receptor? We basically have an endosome with low pH. Since the pH is low, you know, it's acidic, proteins don't really function acidic pH. So what happens is that this receptor won't be able to function, so it basically goes out. I'd be like, I don't want to stay here anymore, so it goes out. However, the mannose is basically allowing our lysosomal proteins to survive in this low pH, right? So mannose allows them to survive. And so with that being said, the enzymes will go to the lysosome, or like some references would say that this endosome is now going to become the lysosome. So is this clear? What happens is, just like the start, you know, we take it from the cytoplasm to the endoplasmic reticulum, we start the modification. The key point here is that we add a sugar known as, mannose with N-acetylglucosamine, and this mannose is going to be either targeted by the Golgi apparatus, which pokes phosphate into it. Mannose 6-phosphate gets picked up by a receptor, which forms an endosome. We take off the two things that the Golgi added, which are the phosphate and the receptor, and we end up with a lysosome. All clear, all good? Here's a picture to describe the process. You got a protein right here. We add the mannose on it, and then we send it to the Golgi. Inside the Golgi, add the phosphate on the mannose. This phosphate is going to be picked up by a receptor. This receptor buds off, and as you can see, once it buds off, you remove the phosphate by a phosphatase, you remove the receptor into the endosome, and you have your lysosome. Finally, you end up with a lysosome. Right? All good? Now, one last point you need to understand here is what happens if you have a deficiency in transport of the lysosomes. So, you guys know what exactly is the function of lysosomes? Can any of you tell me what exactly is the function of lysosomes? Right, yes, exactly. So lysosomes, what they do is they're breaking stuff. This is the way I remember. I don't really go into the depths of it. Whenever someone asks me about lysosomes, the only thing that comes to my mind is lysosomes are for breaking. Okay? So with that being said, what do you think is the function of the enzymes that are sent to the lysosomes? They are going to be the enzymes that help us break down stuff. Okay, so if you can't send these enzymes to the lysosomes, the lysosome is basically just like an endosome. It's a saccule, or it's basically this ball that has stuff inside of it that you don't have the ability to break it down. You don't have the capacity to break it down because there's no enzymes. So it's just like a vacuum holding up this stuff, right? And this disease is what we refer to as a storage disease. Now, one of the types of storage diseases is the eye cell disease, which is also called mucolipidosis type 2, which is also called inclusion cell disease. Okay, so it's basically a mutation in the enzyme that adds the phosphate. And where did we say this enzyme is found? It's in the Golgi, where we're blocking phosphate. So a mutation of this enzyme means that we have no amino 6 phosphate, means we can't take the stuff to the lysosome, and instead, since they're in the Golgi, the endosome doesn't recognize that we need to take them because there's no receptor. And what happens is that the endosome will take this protein that has mannose, which has like a random function, it just sends it outside the cell, right? We can no longer have it being going to the lysosomes. Okay, so with that being said, what happens if you can't break down stuff inside the lysosomes? These stuff will build up, and they are going to be called inclusions. Why are they inclusions? Because they're inside the particle or inside the lysosome. We call them inclusions. And now I added the symptoms that were mentioned in your slide. I'm not going to go over them. You guys most likely don't need to go over them either. Just read them quickly, be like, oh, okay, that's cool, because you're not going to get asked about them in first year. Now, that being said, our last part here is that, so now we understand that proteins can go into the membrane, they can go outside the cell, they can go into the lysosomes. One last point is proteins that are going to be secreted outside the cell. How exactly will they be secreted? There's two different types, constitutive secretion and regulated secretion. What does constitutive means? Constitutive, const stands for constant. So it's constantly releasing, it's always releasing. An example of a protein is collagen. While on the other hand, regulated, it needs regulation. So it's only going to be released when you're regulating it or when you have a stimulus, right? So the only stimulus is received. And what is an example of it? It's insulin. And basically, what is the stimulus for the release of insulin? The stimulus for release of insulin is a low blood glucose level. You don't need to know this detail. Just basically know what regulated is and what constitutive secretion is. And with that being said, we conclude our first part of the lecture, which are the proteins to be sent outside the cell. I'll give you guys 5-minute break now so that you can take that down, break that down. Feel free to ask any questions you have. And in 5 minutes, inshallah, with a fresh mindset, we'll go back into the next part, which is the proteins for the cell. All right. Okay, feel free to ask any questions and feel free to take your break right now. We'll start at 8:05 p.m. inshallah. Tune the recording. All right, so for the next part of the lecture, we're going to be discussing the proteins that are going to be inside the cell. So where we might need these proteins, we might need them in the cytoplasm. And what is an example of a protein inside the cytoplasm? Honestly, you don't need to know anything. That is basically just think of enzymes. So enzymes inside the cytoplasm. You can have them inside the nucleus. Also think of enzymes. You can have them inside the mitochondria. Also think maybe of enzymes or other stuff that you might need inside the mitochondria, right? So now, for this part, it's going to be inshallah a lot easier than the proteins targeted outside the cell because it's just a few concepts that you need to know, and if you get them down, it's going to be easy. It's just basically this entire thing again for every different destination, okay? So again, I brought up this slide so that you can remember our analogy before we go into this lecture because we'll be repeating it again, and I want you guys to really remember it because it helps you memorize everything. So we got our signal on this protein. The signal is going to be recognized by the receptor. So this receptor, this is a person, this is a child, and this is a bully. The bully is bullying the receptor, and then they're walking around. And then what happens is that you got your destination, which sees this receptor. How does it see it? It has another receptor for this receptor. So it sees this receptor, it takes it, and it uses energy to separate them. It sends the bully outside, and it takes the protein inside. And once it's inside, it cuts off the signal that brought this protein, and this way we have a mature protein. Okay, remember this as we're going to be using it a lot. the first thing begins here is the nucleus. Now you can already see a difference from the start of the process, is that translation this time finishes in the free ribosome, right? Because there's no longer the early signal that says to go to the endoplasmic reticulum. So if there's no that signal, you can keep replicating until your protein is produced. So once this protein is made, now this entire protein has the signal, right? So we got the signal. So what is the first step? Signal. What is the signal for the stuff that goes to the nucleus? Has an easy name. Nucleus localizing signal. It says I'm located to the nucleus. So we got the signal. The next thing we'll need is the receptor for the signal. So what do we call this receptor? Nucleus importing receptor, or NIR, or important. What does it do? It imports this protein that was made for the nucleus. So it imports into the nucleus, and this is why it's called nucleus importing. Okay, so we got the nucleus, we got the protein, we got the signal. So the signal is, we got the protein signal, and we got the receptor for the signal. What is the next thing that we need? We need a receptor for this signal, or for this receptor, a receptor for the receptor. And this receptor is going to be found on the nucleus. Now, okay, what exactly is this receptor thing going to be? This time is what we call RAN-GTP. And as you can see, this RAN-GTP is not really, so RAN-GTP right here, it's not really a receptor, but it's just a structure, or this, this particle found inside, and it acts. So let's assume that it's the receptor for the nucleus, okay? To keep our analogy going. So RAN-GTP binds on it. And what did we say was the function of this feature? It uses energy to separate them. And very kind of it, RAN-GTP already has its own energy, which is GTP. So it breaks down the energy and separates them, right? So we got the protein inside the cell, and we got the receptor, which is going to be sent outside. Now, RAN-GTP takes the receptor. If you can see right here, it basically takes the receptor by hand and actually takes it outside the nucleus. But basically, it does the same thing. Uses this ATP to send it outside, okay? Now, one important thing that you need to differentiate here is that basically hydrolysis of ATP here happens only outside the cell. So see, hydrolysis happens only outside the nucleus. So you can think of it, to add our analogy, you can think of it as RAN-GTP sees this bullying process, but it doesn't want to traumatize the protein. So what it does is that it tells the protein, please wait here. It takes the guy outside, and then it starts beating it up. So this is why it takes it outside, it hydrolyzes the ATP and starts beating it up, right? So, and once this is done, I added this, one second. So I added the I guess part here only because in the slides and in the picture, it doesn't show that it removes the signal. So I'm only assuming that what happens next is that we remove the signal and we keep it inside the nucleus, right? So if you use our own analogy that we made together, you can apply it on the nucleus, and the nucleus destination is as easy as anything else, okay? So we got the signal, which is the nucleus localizing signal. We got its receptor, nucleus importing receptor. It imports it to the nucleus. However, once it gets inside the nucleus, the teacher sees them. Who's the teacher this time? It's RAN-GTP. RAN-GTP says, stay here, protein. I'm going to take this guy. I'll deal with him outside. Once they go outside, RAN-GTP starts beating it up. So it uses energy. So it breaks down ATP and sends the receptor away. And inside the nucleus, the protein has its signal removed, and we got it. Is this clear for the nucleus? I hope, inshallah, it's clear, and we can go to the next organ, or the next organelle, which is the mitochondria. Now, before we go to the mitochondria, you can already see a few differences. Now, the main idea is still there. However, there are a few differences. And this is mainly because of the structure of the mitochondria. Now, some general aspects of the mitochondria is that, one, it has two membranes, an outer membrane and an inner membrane. And two, the mitochondria has its own DNA, so it's able of making its own proteins from inside. However, there are a few proteins that the mitochondria can't make on its own, and it needs them from the human DNA or the cellular nuclear DNA. So these few proteins, how exactly will they reach inside the mitochondria? This is what we'll discuss, okay? So keep in mind that the mitochondria can make its own proteins. However, there are a few proteins that we need to take them from outside, and how exactly they go in is what we'll be talking about, okay? And one last general idea that we need to explain is that basically there's two membranes, right? And we said once you get your signal, you get the receptor for the signal, and then there's a receptor for the signal. Okay, so this receptor this time, it is basically going to be a complex. There are two protein complexes for the mitochondria. One is called TOM and one is called TIM. What does TOM stand for? It's basically something with the T, then outer membrane. And what is TIM? Something with the T, I don't know, but it's inner membrane. Okay, so TOM is for the outer membrane, TIM is for the inner membrane. And then you got a bunch of other stuff such as SAM, Maya, TIM22, whatever, you don't really need to focus on them. Just know there is a TOM and there's a TIM. TOM is on the outside to recognize this receptor and the signal complex. TIM is on the inside to recognize the same structures and they are pulling it in. Okay, so this TOM complex and this TIM complex, they're made of two things. They have the receptor for the signal and they have the channel that pushes the signal in. Okay, and one last thing that I want to mention is that TOM and TIM, are close to each other. So once a protein enters through TOM, the only thing it will find in front of it is TIM. So as it's going through TOM, if we were saying like this is the protein, as it's going down through TOM, so it's going through TOM, you got TIM here. So what happens is that it starts going through TIM, even though it might have not completed going through TOM. Okay, so it's passing through TOM and TIM, and then it goes inside I lost the pen, but it goes inside the mitochondria. I hope that made it clear for you guys. Okay, so it starts through TOM, continues through TIM into the mitochondria. Right? And one last thing, I know I said this a bunch of times, but one last thing is, as it's a little bit different and there are two receptors, there's going to be a little bit of a difference in energy source. Okay, so instead of just using GTP every time, this time let's talk about ATP and a membrane potential created by H+. Okay, so ATP is as one energy source, and then the other energy source is the membrane potential created by H+. And since we're at this slide, I'll just mention it right now, but I'll repeat it again later. What is exactly the difference between the ATP and the membrane potential of H+? You find the ATP to help you in TOM. So ATP is found here, while the membrane potential of H+ is found here. And what is one way to remember it? A way to remember is that, the previous processes, they uses GTP, and GTP is found on the outside because there's only one membrane. So this way you can associate ATP to be found with the stuff on the outside. And on the other hand, you got the membrane potential, which is going to be a new part for the new thing that we have this time, which is an inner membrane. So since this time there's an inner membrane, we have the different energy source, which is the H+ membrane potential. And okay, so this is a way to remember it, but like actual theory behind it, why exactly is it H+, is that because the H+ is going to be found around here inside this area. Okay, so upside here, there's no difference potential, so we use ATP. Here, there's a potential that pushes the protein inside here. Okay, so the protein is running away from this potential. Anyways, if these differences are clear to you guys, the mitochondria right now would be as easy as anything else, right? So again, just like we said, the translation starts and finishes in the free ribosomes because it's for inside the cell. What's the next part? The signal. The signal is going to be recognized by a signal recognition particle. All right, so the signal appears. Now this time it's a little bit different. I put it here, but I didn't mention it, or did I not? Oh yes, so here it's the first line. I forgot to discuss it, but basically, as just going on with the idea of this part mitochondria structure is different. We're also going to be using chaperones to help us fold the protein. Why exactly will we be using chaperones to help us fold the protein? Because we need to keep a very flat protein outside the mitochondria. Why do we need a very flat protein? So that it can pass easily through the TOM and the TIM. And because we had it to be very flat outside, we'll also need protein chaperones again inside the mitochondria to help us fold it again, right? Since we didn't fold it at all, we kept it very straight outside. We need some support in folding it inside. Okay, and this is why we need chaperone. Now, with that being said, we got our signal. Now, this signal is going to be recognized by the particle. Now, what is the particle this time? It's the chaperone. It's one of the chaperones. Okay. So I added it between brackets just so that we can use our analogy. But basically, the chaperones are going to be the particle or the receptor that binds on this protein, okay, or this signal. So once the chaperone binds on it, what did we say was the function of the chaperones that are outside? They're basically preventing the folding. Okay, they prevent the folding so that it can stay straight. Now, once it binds on it, what happens is we prevent the folding of this protein. And we got our receptor and protein. They're walking around, are recognized by the receptor for the receptor. What is the receptor for the receptor? It is TOM. So once the receptor for the receptor binds on it, what does it do? It uses energy to separate them, okay? So we're gonna use the energy. And what did we say was our energy source for outside? It was ATP. So we use ATP, we separate the chaperones, and we take the protein inside, okay? So we use ATP to remove the chaperone, and we take the protein inside. And remember how we said, as the protein is going in, what happens? It talks to Tim as well. So Tom is pushing it in, and now Tim takes it, and Tim continues the process of it, okay? And one last point, how did we say Tim moves it inside of it? Tim uses H plus gradient as energy. As you can see, here are the H plus, they're inside or between this membrane. Okay, so this first step is clear to you guys. So we got the receptor, we got the protein. The receptor on it is a chaperone to prevent the folding so that it can stay straight and pass through the tool membrane. And then this is going to be recognized by its own receptor. So its own receptor is going to take it, which is a TOM. It uses ATP to separate them apart. And as it's passing in, it goes through Tim. And Tim is going to be using H plus as the energy to get it through. Okay. And once we're pushing it through, what happens? You got your protein inside. You cut off the signal because you don't need it anymore. You have the protein inside the cell. You get this chaperone. Okay, so this part is a little bit different this time here as well. So we said we need a chaperone inside the protein in order to fold it, right? So we said we need a chaperone inside to help fold the mitochondrial protein because we kept it very flat outside. So not only does the chaperone, so HSP70 is a chaperone, remember? I don't know why it's the same name here and there. I don't know if it's the same particle, it's different. However, they're both a chaperone, they're both called HSP70. And what does it do? HSP70 not only helps fold it inside, but also it uses ATP to pull it in faster, okay? So it pulls it in using ATP. So as you can see here, HSP70 helps pull it in down more, and once it's in, it helps fold the protein and remove chaperone again, and you got your protein full and ready and complete. How does that sound good? Is it clear to you guys? And once again, I'm gonna do this again in order to remove the background noises that are happening. Sorry about that. Okay. So yes, is it clear to you guys? Is the mitochondria clear? If you understand the mitochondria, inshallah, everything, it's probably the toughest one because everything is different than the rest. But if it's clear, please let me know, and inshallah, we're done with the lecture, basically. So again, what happens is you got your protein, you got basically, so you got your protein with a signal. The receptor is an HSP70. It keeps it straight. The receptor for the receptor is a TOM. TOM uses ATP to push it in. Inside, it's going to be interacting with TIM. TIM uses the H plus energy to push it in at the start. Once it's in a little bit more, you cut off the signal, and then you got your second chaperone, which is an HSP70. It uses more ATP to pull it in down faster, and it is also going to be helping in the folding process. Okay. Now, the fourth place you have is peroxisomes. Now, there is no much details that has been mentioned in your lecture. The only thing you need to know, you got your protein completely folded and produced. Where is it going to be produced? It's produced inside the free ribosomes. So the protein has a signal. The signal is going to be binding on by the receptor, and then the receptor takes it into the peroxisomes. Okay. So what is the signal? The signal this time, remember at the first slide, I told you two important things to remember. The N-terminal hydrophobic sequence to the endoplasmic reticulum, and the serine, lysine, lysine that's going to be used for peroxisomes. Okay, so this is our signal. And what are our receptors? They're called peroxins or PEX5 and PEX2. So you got this signal, you got your peroxins, PEX5 or PEX2. You don't really need to know this. You don't need to know the details. Just know that the signal with the receptor pushes it inside the cell, right? Now, one other thing that I added here is basically what exactly is the function of peroxisomes. So peroxisomes are for two things: lipid metabolism, or the breakdown of lipid, which is fats, and detoxification of ROS. What is ROS? ROS is reactive oxygen species. Now, reactive oxygen species are basically there to help your body break down and fight off stuff. However, if you have too much of them, then this is going to be bad. So you don't want too much of them, and you don't want too little of them. If you have too little of them, you're going to have to make some more. If you have too much of them, you have to use the peroxisomes to remove them. Why? Because if you have too much of them, and we said their function is fighting, they can't really differentiate a bad thing and the good thing. So they can't really differentiate between fighting the bad stuff or your actual healthy cells. So if you have too much, you're going to start fighting your healthy cells, and we don't want that. Okay? With that being said, and if we understand that their function is for lipid metabolism and breaking off this reactive oxygen species, what do you think will happen if you don't really have functioning peroxisomes? All right, before we go into that, so remember how we said the lysosome takes in the proteins that are enzymes? The peroxisomes has a kind of similar function. So what do you expect the proteins that it's taking it in? They also have a kind of similar function of breaking down stuff, okay? So if you have a deficiency or a defect in your peroxisomes, so maybe it's the process of transporting these proteins. So you don't have really these enzymes, okay? So what happens here? You have a disease, okay? This is known as a Zellweger syndrome. I don't know, try to find a way to remember it. It sounds weird, Zellweger syndrome. So what happens in Zellweger syndrome? You have a mutation of PEX2, and we said PEX2 is the receptor. So PEX2 is a receptor for the proteins. And so what happens is that you end up with this empty peroxisomes, and this leads to problems in your brain, maybe in your liver, maybe in your kidney. And since it happens mostly in children, you can have a child dying soon after birth because it's a really dangerous disease. Okay? Is it clear to you guys? I don't think there's much to be said here. Pretty straightforward. So let's go to the next thing. All right, last and final thing we have. This is not really protein targeting. However, it is basically the final point we need to know. And now we need to understand the following, okay? So we said that sometimes, proteins have their signals. We take them through their signals, and then through their signal, we send them into their destination, right? Now, sometimes this process can be wrong. Okay, so the process of transporting the proteins can be wrong. Sometimes your body produces the wrong proteins, Sometimes the proteins have the wrong structure. Sometimes the proteins are not really effective. So anything you see, the common theme here between all these four situations is that basically you have the wrong proteins. Okay, so if you have the wrong proteins, what does your body do? If you have the wrong proteins, just to keep it short and straightforward, we don't want you. We don't want the wrong proteins. So your body tries to break it down. And what does it use to break it down? It uses a proteasome. So a proteasome, you see the protea kind of starts for the protein at the start, and proteasomes are zoming and breaking down the proteins. Okay, so proteasomes are breaking down defective protein. Now, what is the signal for the proteasomes? It is ubiquitin. Okay, it's called ubiquitin. Now, why is this a little different? Because ubiquitin is not actually going to be a part of the DNA sequence. Because when you are making something, you're not making it to be bad, right? It just happens that it is bad. So the protein is made, however, it turned out to be bad. So your cell be like, okay, this is bad, get a ubiquitin placed on you, okay? So the way I remember ubiquitin, so ubiquitin is always, please, whenever you see ubiquitin, it's going to be associated with destruction of proteins and proteasomes. Please, whenever you hear the word ubiquitin associated with proteins, associate proteasomes, and defective proteins. The way I remember it is that, so I used to play a lot of video games. So ubiquitin, so kind of ubiquitin. So you're quitting the game because you're bad, you're dying. I don't know, I just used to play a lot of games, so it helped me. So ubiquitin because you're bad or whatever, okay? So you quitting a lot. Anyways, so remember how we said that it's not a part of the protein and we need to put it on the protein or tag on it? We use something known as ubiquitin ligase. And if you speak Arabic, it's really easy to remember ubiquitin ligase because ligase is very similar to the word tape or zig. So ligase, it's taping or placing it on the protein. Okay, is it clear? Now, one last part is basically what is an example of a disease where you're going to be using your ubiquitin? It is cystic fibrosis. So what happens in cystic fibrosis is that you have a mutation which causes an abnormal protein, and so basically you have an abnormal protein, and so you are going to be using ubiquitin in order to destroy this abnormal protein. Basically, this is what you need to know about it. Now, I'll go into a little bit of extra details here. What exactly is the mutation that happens in cystic fibrosis? It's a 3 nucleotide delete of, or 3 nucleotides. Once you remove them, you removed a phenylalanine, okay? Phenylalanine. Now, if you see these, you're going to see them in every mutation. What does a triangle mean? Triangle means mutation. What does 508 mean? It means it's position 508 of the protein sequence. Now, what does the F mean? If you remember in your amino acids lectures, so amino acids can be referred to as by their name. So we can refer to them as phenylalanine by 3 letters. I don't really remember what they were for phenylalanine, but some might say PHY maybe. So 3 letters, PHY, and you got the 1-letter presentation. So here we use the 1-letter presentation. So the F is for phenylalanine, the 508 is for the position, and the triangle is for the mutation. Okay? Is it all good? Is it all clear? All right, I hope it's really clear for you guys. And with that being said, we're done with the lecture. Please let me know if you have any questions. Please feel free to ask your questions. If you don't have them right now, and if you're studying the lecture later, you can feel free to text me for your questions. And we'll be doing some questions right now. All right. Now, one thing that I want to do just before the questions. So for cystic fibrosis, I remember that we mentioned it in a previous slide. So I'm gonna go back to that so that you guys can create links. But if I can find it. Okay, I didn't find it. Anyway, so yeah. Anyways, forget that. Let's go to the question. All right, let's start with question number 1. Who would like to answer? Please feel free to write in the chat. I'll read the question, or if any of you would like to read the question, you can read the question. And please type your answers in the chat. Okay, so the intended final destination of a new protein is within a peroxisome. However, the peroxisomal targeting signal is not properly incorporated into the protein. What will be the final destination of this protein? I'll give you guys a second to answer that. So what will be the final destination of the protein? Okay. So yeah, basically most of you guys got it right. The answer here is the final destination of the protein will be the cytosol. Okay. So why is it going to be the cytosol? Because we said the protein is going to be for the peroxisomes. And where will the proteins of the peroxisomes be made? They're going to be synthesized in your free ribosomes, right? So they're going to be made in the free ribosomes. So if they're going to be completed in the free ribosomes, which are found in your cytoplasm, once they're made there, we need a signal to move them to the peroxisomes. However, there is no signal. So what happens is that they're going to be sitting around there inside the peroxisome until they get broken down, until they get, whatever else happens to them. But basically, since they're completely made and sitting there, they're going to be sitting inside the cytosol. Which brings us to the second question, or question number 2. A precursor protein, which is intended to function within a lysosome, fails to receive a proper lysosomal tag. The protein will be sent to, or when exactly will you find this protein? All right, I'll give you a second to guys take a guess on this one. Right, so yeah, as you guys said, the answer here is gonna be E, outside the cell. So why exactly is it gonna be sent outside the cell? Because it was supposed to be for the lysosome. And remember, where did we say the lysosome tag takes place? The lysosomal tag takes place inside the Golgi apparatus. Okay, inside the Golgi, we poke it with phosphate. And once it gets poked with phosphate, we know to send it out. However, it's inside the Golgi already. So it already started in the free ribosome. And then the free ribosome saw the signal. The signal took it to the endoplasmic reticulum ribosome. And then from the endoplasmic reticulum, we sent it to the Golgi. And right now we're inside the Golgi. If it doesn't get sent from the Golgi to the lysosomes, then most likely the Golgi will send it outside the cell. right? Because the Golgi is programmed. You either go to the lysosome if you have your tag, you either go to the cell membrane if you have another tag, I guess, I'm not sure, or you go outside the cell. So once it's in the Golgi, if there's a no lysosomal tag, it's going to be sent outside the cell, right? So this is why it's here. Now, next question. A 4-month-old male is evaluated for muscle weakness and poor muscle tone. You do a physical exam and you can see hepatomegaly. What does hepatomegaly stand for? Okay, never mind, it's there. And further testing reveals the presence of heart defects. Excess glycogen is found within cells of his muscles, heart, and liver. Deficiency of an N-acetylglucosamine 1 phosphotransferase is suspected. Based on this information, glycogen accumulation has occurred in which location of the affected cells? Right, I'll give you guys a minute for this one. Okay, correct. So the answer here is C, lysosomes. So basically, if you just read the name of the enzyme, you can answer the question, which is basically phosphotransferase. And we said there is the enzyme responsible for packing phosphate onto the proteins to be sent to the lysosomes. So, this is a previous question. Now, I just want to add up why exactly this glycogen accumulation happened. Is that, remember how we said every protein that was sent to the lysosome is responsible for breaking down stuff? If you can't send this protein to the lysosome, if you can't really break down this stuff, what is going to be happening? You're going to have them building up and accumulating, and this is what happened with this patient here. And this is the last question that we have. In a patient that has cystic fibrosis, you have a mutation, and basically the protein falls incorrectly. And basically so, the protein is attached by ubiquitin. Whenever you see ubiquitin, what is the first thing you want to associate it with? Breakdown of proteins, okay? So what is the fate of the CFTR? Look at the choices. Is it secreted from the cell? No. Targeted by lysosomes? No, because there's no signal. It's repaired? Not really. Basically, it is degraded. I don't want to go through the choices because we must remember immediately, whenever there is ubiquitin, it is the breakdown by proteasomes. And with that being said, I'll go to my last slide, which is a few resources and books that you guys can use for practicing for your exam. And please scan this QR code and fill up the survey. And with that being said, we have concluded our lecture for today. Inshallah, I hope you guys benefited from it, and I wish you the best of luck. You can find my contact number at the first slide. I'll be sending it out to you guys now. You can find my contact number in the first slide, so feel free to text me for any questions that you have. And thank you guys so much for attending this lecture.